We're talking about actually what I think is a more natural process of science that we just don't embrace enough, which is I might get it wrong. And that's because the process is about not trying to get something right. It's about trying to get something less wrong. And that's a really interesting concept to think about. And it's really hard to wrestle with. So yeah, even in our papers, not to feel it whole, like it's not trying to make a general claim or a specific claim to an individual paper. It's trying to say, listen, if I can replicate something again, that just means I increase my confidence. Yeah, that's probably really good. And if I can't replicate it again, it doesn't mean the original is wrong. Could be. It probably means we just don't know enough, right? I couldn't pick it up. I couldn't take somebody's finding that they published, have somebody do it again. That just tells me something's missing. And it means I don't have as much confidence as maybe I should, so I should take pause. That's at least that's the approach that we take. And that's really important because that means you can calibrate how to use evidence, because you are just changing your confidence over time. And in the end, you're always going to have to make some policy decision with whatever evidence you have, right? COVID's a great example of that. But to me, it's always like, but don't you want the best evidence at that best moment in time, like whatever it is? So replication to me is like a huge tool that we need to use more in research to be able to have more confidence in our findings.
Hi, my name's Lester, and welcome to another episode of What's with RWE. As you can see, we don't have our co-host again. Costas, unfortunately, got called away, and he's on the road right now. He really wanted to dial in, but unfortunately, he has absolutely no reception. So the fortunate thing, I guess, is I get to commandeer this entire conversation with another lovely guest today. We have Tim Errington on, and this has been a long journey. I listened to a podcast where Tim and his team were on it, and I very much wanted them to come on What's with RWE. So Tim is the Senior Director of Research at the Center of Open Science, and he actually led a project called the Reproducibility Project for Cancer Biology that he'll talk a lot more about during this podcast. He has an MA from UC Berkeley in molecular and cell biology, actually myself as well, actually molecular cell biology, and a PhD. So I don't have a PhD, but he has a PhD in University of Virginia in microbiology, immunology, and cancer biology as well. So Tim, welcome to the podcast.
Awesome. Thank you. Thanks for having me.
Thanks a lot for joining us. So maybe let's start off with because a lot of people may not be familiar with your organization, Center of Open Science. So maybe you can explain a little bit about Center of Open Science, the Reproducibility Project, and why it's very important, right? I think it's incredibly important work that you do, but maybe you can explain why you believe it's important work.
Okay, that's a lot. I'll go at it. Interrupt me. So, the Center for Open Science, I'll just want to say COS if that's okay. We'll just abbreviate it. So we're a nonprofit that is based in Central Virginia in the United States. So we're about almost 11 years old, a little over 11 years old. And our mission is to increase reproducibility, integrity, and openness of academic research. And that's agnostic to discipline. So, you just heard my background, right? Preclinical cancer biology is my focus. But our founder, for example, Brian Nosek, he's a social psychologist by training. So I love it because it's looking at it across disciplines. How do we go about that mission is there's kind of like a couple key activities we do. One that I'll talk about, which is the research that I've done and my team does, which is meta-science research. So research on the research process. I'll get back to that in the Reproducibility Project and why we did it, why we think it's important, and why we keep doing it in other areas. We also do a lot of policy work. That policy work is really more focused at the incentive system. So if we want to, increase openness, reproducibility, we need to change the incentives. So that looks at journal policies, funder policies, institutional hiring and tenure policies. These things all impact the type of research we do. If we just reward publications, which is largely what we do, and not rigor and reproducibility and openness, right, you get a very different culture. So we want to shift that, have those incentives there, but reward the behaviors we want, openness and rigor. We also need to enable those to occur. So we do a lot of technology development, open source tool development to enable openness to occur, and other practices we think that are important. One that we advocate for, I think, is brilliant. Little bias there, of course, which is pre-registration or registration of research. So we do this in clinical trials. You map out what your protocol is before you begin recruiting patients. Ideally, you do it prospectively. That's really helpful. It lets you know what's going to happen. Maybe they don't succeed. Regardless, all the results are out there, and there's a lot more rigor because it's about a plan design. How many patients should I have? What do I expect to observe? We're trying to do that into other areas. It has huge success in the clinic. Still more to do in clinical trials to improve it. We're trying to pull that into the preclinical space. I think that can actually give us a lot more insight into what is and is not working in the preclinical space to make it a lot stronger when we move things into human studies. So I think that gives you a big overview, high level of what we're doing at the Center for Open Science. So let's zoom back into the arm of the team that I work on, which is the meta-science team. So we largely do a couple different activities when we're trying to investigate, research practices. Some of the work we do is trying to understand norms and behaviors, and some of it is trying to test different interventions, different ways of improving openness and rigor. But one of the things I think that we're really well known for, and that is actually foundational to get to those second two parts, is what's the current state of affairs right now. So how do we conduct and share research? And one of those areas that it took us about 8+ years to finish it was in my background, preclinical cancer biology. So when we first started the center, shortly after that started is when we started that project. So way back in 2014 is when it started. And the reason we started it is, and the reason I think this is an important topic, is right now, I think largely you can take or think of the published literature at face value. It's been published, I must trust it. I think we all hopefully are learning that's not actually the case, right? The pandemic, I think, did a great job of showing that, which is, things get retracted. We get things wrong. Maybe not all the information is there that we want. So it basically means it's a much harder. You can't just say, published equals scientific truth. You have to say, well, published just gives me information. I still don't know how reliable that information is. There were also concerns, and there continues to be concerns, around how reliable it is from other actors. So in the preclinical space back in 2014, if we go back in time, the pharmaceutical industry was a early kind of whistleblower, if you want to call that, in terms of saying something's not right here. Bayer, Amgen are the two big papers. They wrote these white papers saying, listen, we pick up the academic literature, we try to take it into the pharmaceutical area to turn it into, hopefully, therapeutics, drugs, right, treatments that will find its way all the way into the public domain, something that will actually help lives. But what we find out, and again, it's more retrospectively, it doesn't happen all the time. In fact, they were giving numbers like 11, 25% of the time could they replicate the results, the rest they could not. So that's really crazy when you think about it, right? Take those numbers at face value. All the research we produce, are we saying that we produce, right? Are we saying that we can only replicate, only use about 1/10 to 1/4 of it max? It causes pause, right? The reason we did our project is that's all they said. They just said, hey, we can't replicate these results. We only see these low percentages. And again, that caused a huge alarm for good reason. We came in and said, okay, well, that's an interesting question. I wonder if that's true. Let's do it. Let's do it in the open, though. Let's make sure that all of our research is in our replication done transparently. We'll basically model what we as an organization are practicing and trying to encourage, which is openness, rigor, reproducibility of our own work. The reason we want to do it in the open is it's easy to give you a number. It's much more important, I think, to tell you why that number occurred. So when we started this project, that was actually the intent from the beginning. We're going to open everything up. We're going to have the entire process, the selection of papers that we did, every step along the way, we're going to make that as open and transparent as possible. So that way, instead of just having a number at the end, we're actually going to say, hey, look at the evidence itself. I think that's just as important. I'll jump real fast. 8 years later, right? Plus, I'll give you the top line. I used to always hate giving this, but I'm comfortable doing it now, which is it's almost like a coin flip in terms of how many can replicate. But there's really there's two stories to tell here. One was it took a long time to try to replicate research. So we can get into that more if you want in terms of just the challenges we had. But of the ones that we could replicate, which is, not nearly as many as we attempted, is 54% on using various different metrics of replication success. So to me, it's okay, well, that's interesting. That's basically the state of affairs. The state of affairs is it's really hard to get information from published papers and asking authors. And when we do get that information, maybe half the time can we replicate it. So I think to me, this is wow, there's so much more potential that we can do here to improve the research enterprise and get a lot more out of the investment that we are rightly so putting into, this space.
Great. No, thanks for laying that out, because it definitely very, very big topic. Of course, we want to dive into each one a little bit more deeply. I just okay, this may be definitely an somewhat off-putting maybe question, but I'm going to ask it anyways. Is are you guys just not the most popular kids in the? You must be not super popular amongst the academics. Hey, let me try to reproduce your work,
Yeah.
right? So how do you like what? How do what is the
Hmm.
reception of the community? And of course, there's definitely people that I'm sure back you and are very very supportive of what you do, but how about the general community? What is their appetite for what you're doing?
That's a great question. Yeah, I would say, yeah, sometimes don't make friends. So there's a way to get past that too. Like the approach I've always tried to model, right? And we do this in other projects as well in different disciplines is it's not about we're not trying to point the finger at anybody. It's not about a blaming. It's just really saying, listen, this is what was a cornerstone of research. We should be able to take information and have somebody independently do it again. Because if not, then what are we doing, right? What are we doing in this prize? At the same note, you're absolutely right. People get really upset with us. Um, so, right, it's like try to take that high road as best as possible, and it doesn't. And it's totally human nature, right? It's like, Tim, I can't believe you're doing it. I would do. If somebody came to me and was like, I'm going to replicate your results, I'd be like, whoa, what are you doing? Why are you doing that? The big thing that we try to do is our study was never about like I said, pointing fingers or trying to say there was a reason. We did it because we're like, that's a really important paper. Lots of people are using it. They're reading it. They're citing it. That's awesome. So let's see if we can replicate it, right? I think the response kind of changes over time, and it depends on a bit who you want. I think early on in the project, for sure, there were definitely people that were glad we were doing this. And the reason why is because of what I just told you, the rationale, which is good. You're doing it in the open. You're doing it the way it's supposed to be done. It was easy to say a number. It's much more important for us to show our evidence. Okay, so I think we got some people on the right board. At the other time, I thought some people were like, no, the way you're doing this is all wrong. You're basically, this is not going to help science. You're kind of airing dirty laundry. Like, this is not good for what things are doing. All the way to people saying this is a silly idea. It doesn't matter what you find. You're never going to make a difference. So I got the full spectrum before, but it does kind of change over time. And there's an example by even looking at other disciplines who have wrestled with this. And I think right now we're at a stage where there's probably still some people, and it's easy to not find them. I actually do appreciate hearing it, which is still thinking, this doesn't make a difference. But I think now it's kind of more of an acknowledgement like, yep, okay, yeah, we're not as replicable as we should be, not as reproducible as we should be. What are we going to do about this? And that's actually completely changes the tone. So before you just get like your champions, the people who've kind of already appreciated it from a different venue of, yes, there's something we should do. I think that just takes took almost 10 years to get to what I'm saying now. If we look at psychology, just like we'll use another discipline as an example, because that's one that went through a similar project that Brian Nosek, our CEO, led. They had the same issues, the same confrontation early on. Now they're a little bit farther along in their journey than we are in the preclinical space. And I can tell you right now that it's much more normative to talk about it and even do replications. So the tenor changes completely to where the culture almost is saying, okay, yeah, we can do this. And I kind of almost expect people to do it. I think that still requires the whole system. Like that's why that incentive piece is so important, right? Which is it's you still have to reward it in order for people to keep doing it. They're not going to do it on their own behavior. But yeah, it's a really good question, and I still there are times that like I said, I still know that there are groups that are just not still happy to hear about this. But I think hopefully the time has gotten to let's just accept that this is an issue, and now what are we going to do about it to make our research more rigorous?
That's good that you're slowly moving people towards that, not pointing fingers, but very much trying to improve things, because that's really ultimately what the community is looking for. And just for like folks that maybe are not they don't do science in terms of they're not scientists or not researchers, but they're just every day they go they're mechanic, they work in office. Like, why is that important? You already touched on a little bit of it in that, hey, you know what, one of which is a lot of tax dollars go into this type of research, right? So why is this such a big such a challenge that's worth solving, at least from your perspective?
Yeah, and there's a lot of reasons, right? Yeah, and I think you're right. Like one simple one is that public good, right? A lot of this is public good funds. It should go back and become public good. Accessibility of information is actually kind of crucial to this, right? I think there's another reason I think this is important outside of that argument, which is that we need to make sure that we have a process in place that essentially means you shouldn't trust who did the research, right? It's not about like the name of the person or the journal it's in, right? Like you trust research because like it constantly keeps interrogating itself, right? So this process, this scientific process of basically saying like, it's not about getting a right or wrong. It's about trying to say, I think I'm more right now. And really it's about like, right, it's reducing uncertainty. If we don't embrace this, if we're not really open with what we're doing, if we can't do more replication research, it makes that process really hard. And I think it makes it really hard for the public to be able to like trust science, right? Because like it's not about, it's really like trusting that process. I'll give you an example of like in a different space of like where I think this is really important. We could do COVID if you wanted to, but I'll actually go back to like mammograms, right? There was like a recommendation on changing the age of when mammograms would occur. And I think a lot of people got very confronted of like, why would you change that? Like, why would the recommendation change from one age to a different one? And that's because research is constantly updating itself, right? It's constantly saying, I don't think I'm right all the time. So I'm just going to keep taking more data, right? Or keep looking at the same data again to see if I can kind of like improve that to put it into practice. And that might mean telling the public, hey, what was right, or what we thought was quote unquote right 10 years ago is not right anymore. So to me, like when we look at replication, we're actually doing the same thing. We're trying to say, listen, there's a lot of information out here. Don't take it at face value. We have to keep beating it up. Like we just don't know. So I'll go one more thread back into that argument of economics. To me, in the preclinical cancer biology space, to me, this is like super important because we're moving from all this research, this investment in basic research and translational research in an attempt to get something out into the public space. And you probably see headlines all the time. Oh my gosh, this new study in shows X, right?
Yes.
And that's great. Yeah. I think it's awesome to have that excitement. But I think like the research we've done and like what Bayer Amgen is trying to say is like, yeah, but like hold off, like just wait a second. And I think it's also back to like not just investment of dollars, but also like that hope, which is I get really, really upset when I see us run to first in human too fast. It's not because we shouldn't, right? These are really, important things that we're doing, diseases that we do need to find some way to make life a lot better, detect it earlier and figure out ways to get closer to a cure. But I don't think running towards first in human trials is the way to do it. I think instead we need to fail earlier in the pipeline, make that more open and transparent, and put our resources, because it's always limited to something better, right? So if I have a study and I can't replicate it, it doesn't mean that it's wrong. It means that maybe I should not run to first in human. Maybe I should sit there and say, maybe I should spend more time on the bench doing something else before doing millions of dollars and including human lives into this. Because like my worry is just like, that rush actually can kind of like cause a lot of like, mistrust and misplaced hope. And it's like misallocation of resources as well.
Absolutely. I mean, and you're, just to be very clear, you're not suggesting through the publication, the work that you do, is you're not suggesting that 54% of papers are fake, right? That's just to be clear, so everyone's clear about that.
Yeah, let's make this really clear too. Yeah, let's also call out the specific papers we looked at, right? Like we're not saying any of those are fabric. That's a completely different story. So there's like two narratives here we can talk about. One that we did not touch on at all in these projects. We tend to not, as an organization, really delve deep into it. There are others that do, and thank goodness they do, which is fabrication. Right? There's been recent stuff at Dana-Farber, for example, right? That hit the news. Probably people found it. That stuff always confuses me, right? Which is like, oh my gosh, like you're in a field trying to generate knowledge, and yet somebody's kind of fabricating it. That's really crazy to think about. That's not what we're talking about, because that there's an intention there to like deceive. We're talking about actually what I think is a more natural process of science that we just don't embrace enough, which is I might get it wrong. And that's because the process is about not trying to get something right. It's about trying to get something less wrong. And that's a really interesting concept to think about. And it's really hard to wrestle with. So yeah, even in our papers, not the field at whole, like it's not trying to make a general claim or a specific claim to an individual paper. It's trying to say, listen, if I can replicate something again, it just means I increase my confidence. Yeah, that's probably really good. And if I can't replicate it again, it doesn't mean the original is wrong. It could be. It probably means we just don't know enough, right? I couldn't pick it up all. I couldn't take somebody's finding that they published, have somebody do it again. That just tells me something's missing. And it means I don't have as much confidence as maybe I should, so I should take pause. That's at least that's the approach that we take, and that's really important because that means you can calibrate how to use evidence, because you are just changing your confidence over time. And in the end, you're always going to have to make some policy decision with whatever evidence you have, right? COVID's a great example of that. But to me, it's always like, but don't you want the best evidence at that best moment in time, like whatever it is? So replication to me is like a huge tool that we need to use more in research to be able to have more confidence in our findings.
Yeah. Just for the audience, just so that they kind of understand in some ways, like how does this tangibly, practically even happen? That like, oh, you did something and then it doesn't, you can't replicate it. Like, what do you mean, Tim? So at least in my experience, I'll give you my example, and then you can. I'm sure you have other examples that you can give as well that would be helpful for people. But for instance, for myself, when I used to be a many years ago a pseudo chemist, I would we had to put these DNA molecules on these, functionalize them on these nanorods, right? These really, really tiny structures that were made out of gold. And we followed the publication. And just for the life of me, I couldn't get it to work. I just couldn't get it to work, right? I'm like, I tried like maybe slowing things down, changing these parameters, changing those parameters. And what ended up happening was my colleague had a chat with the postdoc or the PhD student at that time at a conference and said, "Hey, my buddy's been trying to replicate this. Like, why? Like, can you give him some tips? Why isn't it working?" And then the person said, "It works like 1 out of 10 times. Like this method." I was like, "Oh my goodness, that's exactly how often it works for me as well." So just it's these nuances. So it's not that they fake the data; it's just that it's incredibly hard. They don't like tell you that extra little bit. Oh, it only works 1 out of 10 times.
Yeah, that's a good example too, by the way, because like to me, that's really important to know. Not just because, oh, right, I can calibrate my expectations. It's also like, what do I do with this? Because it's only working 1 out of 10 times. Like I can't. It's not something that I can itself repeat constantly. So I think, yeah, that's definitely a big piece of it, which is, and there's a term that we use in this, the field uses, right, which is selective reporting, right? So in that case, somebody selectively reported the 1 time out of 10 that it worked. That's really not desirable. It doesn't mean, again, that you can't repeat it. Clearly you did. It's just that crucial information, that really, really important nugget was gone. The other thing that we see that's really common is, and we can use, your example again, which is a lot of the time when you look at the published paper, does not have the detail that you need it to have. And I think that's like there's no intention here, or at least I'm not going to infer any intention. It's just kind of like the state of affairs, which is it's really weak on details. And a lot of times it'll say standard procedure or as done previously. And you go to this reference and I've done this. You go down this train, you can be like 20 years in the past and you're like, is that really what you did? The same thing from 20 years ago? I don't think so. The reason I think we lose this is, one, we're super focused on the method and on the results and not the methods, even though the methods are crucial to the results. So that means that like the detail, that hidden, that, the devil's in the detail, that the detail is there, but it never popped out. And if you don't share it, no one knows. But the reason you don't share it too is you might not know it matters. So when we do these experiments, right, they're incredibly complex, especially when you do an animal study. So many factors at play and so many that you do not want to have as an impact. So, right, does the, time of day when I do the experiment, does it matter? I hope not. I'm doing a biochemical experiment. Time of day should have no matter, right? Does the humidity in the room matter? No, it should not matter, right? But there are these natural fluctuations we don't measure. And so we don't capture these details because we don't think that they matter. Or I say, oh, just spin the tube. But really what you did is you flicked the tube. And you're like, I don't think, it doesn't matter. Who cares? But a replication confronts that instantly, right? Because it's saying, okay, well, you told me it doesn't matter, so I'll do what I want to do, right? I'll super spin the tube because you told me it doesn't matter. And then you get a completely different result. And again, I think this is why it's so hard, is because we don't want these details to matter. But when they do, then we have a problem because now we're going to have to confront ourselves. The part that I find more interesting, right, is like you get this all the time, especially in biochemical experiments and almost anything, chemistry experiments, where there's tons and tons of moving parts. And you're like, okay, well, should I do it all exactly the same every single time? And I'd say, well, yes and no, right? Like, does it matter from a biological standpoint is the question I have in these cases that we were doing, right? Which is, if it doesn't matter, if flicking, spinning, if I can't have a biological reason for it, in theory, yeah, a replication, somebody doing it independently, like yourself, like that's actually very helpful. It confronts that. There's no reason this should matter, so why does it matter? Which means when you replicate the result, that means there's tons of variation, and that gives us more confidence in the finding. Cool. I specified the details I thought were necessary. Looks like I got it right. We can keep moving up that translational pipeline. If you don't specify all these details and somebody says, I can't get the same result, and you're like, oh, you know what? It turns out it was because you have to flick it 2 times, right? Then you might say, oh, cool, I got the same result. But then you have to ask yourself, why does flicking 2 times matter? Because if I don't have a good reason for it, like what's going on, right? What am I creating here? Because you can get these artifacts, and we're terrified of that, right? Nobody in science wants to have their result due to a lot number, a specific, formulation. It's like, oh, only that one supplier. It's the only way I get the result. Like, you don't want that. But sometimes that happens. And so we need to kind of confront ourselves constantly. And I think it's just, unfortunately, like that's kind of the state of affairs we're in right now, which is the methods are so short that we're missing so many details that, whether it's, flicking the tube, or whether it's just saying use standard procedure, and you're like, there's 20 different standard procedures. Which one should I use? I think that's to me like the low hanging fruit, which is let's do a little more specification of what did you actually do, and just share with me what you actually did, so that way I can explore that if I'm trying to do it independently. Just like you, like it's like, okay, cool, you did it 10 times. Can you show me the other 9 that didn't work, just so I'm aware of what I'm going to get in case I do it myself?
Yeah, no, absolutely. And then you bring a great term like doubles in the details. Actually, now harping back to something that you said just when you were talking about the project itself, and you talked about potentially some of the challenges. I want to actually go back to that and pull on that string a bit. What were some of the challenges with? Actually, you do lay out. I have to kind of give a quick plug. Is that if and anyone finds this conversation very interesting, definitely go to the Center of Open Science to look this up, right? Which is www.cos.io. And then if you go /RPCB, which is the I'm going to have trouble saying this. I don't know how you do. Reproducibility Project. There you go. For cancer biology. You actually lay out like a lot of the different, basically all those metrics in terms of like where things kind of fell off, right? And an example would be kind of interesting is the key reagents, right? That you lay out there. You also said how helpful was the original author as well, right? So maybe you can talk about some of the challenges that it seems like you lay out actually on your actual webpage.
Awesome. Yeah. And there's links to the actual papers that are doing it. So there's, when we think of like when we summarized that all these findings, years of work, we did everything. We summarized it in 2 papers. So I'm going to context before I get to your question. One is what did we find when we could replicate stuff? And welcome to go there. The high level being, like I said, it's like a coin flip, 50%. There's more details in there though that we can definitely get into. But the other paper, the reason we teased it apart is the story that I think is actually the more interesting one is this challenges paper. And so we looked at kind of reflecting on the entire process of, well, why couldn't we replicate all these papers and it was just not a lack of knowledge, to be completely honest, right? So examples, not everyone was sharing their data. Now, it's not a barrier, right? When you don't share, only at 2%, by the way. That's like, just to make that real crystal clear, 2% of papers shared their data, like openly. So, and then when we asked them, only like another like 15% or so did it. I don't know the exact number, but it's like sadly very low. You can get past that. You don't need the data per se, but like it makes it really hard when you're trying to replicate something. If you're like, okay, well, how many mice or how many replicates should I do in my replication? That matters. I have to base it on how big of an effect you found. If you found a super big effect, then yeah, I probably don't need as many animals. That's good. Or I don't need to do it as many times. If you found a really small effect, I probably need a lot of sample size. I can't do that unless you share your data. Same thing with like, analysis. Very few analyses were shared. But yeah, you call out like where it starts to really become problematic, which is these key reagents. So key reagents here would be like mouse models, antibodies, plasmids, cell lines. That's like the workhorse of the molecular cell biology space. And a lot of the times we couldn't get them commercially, or in my preference, right, just my bias would be in an open repository. There's plenty of them out there. We couldn't find either. Basically, the only way you could get it is if I email the author. So of course we would, by the way, right? Because it's like the only thing you can do. You say, okay, well, I can't get it unless I talk to you. So can you give it to me? And what's interesting there is sometimes they would say yes, sometimes they'd say no. And even if they said yes, sometimes they still would say no, which is, like, again, not a great way to make friends, right? Like there was one time we had to intervene. We had to get the publisher and the university involved. And that's not fun, right? It becomes basically a legal process. And we were like, oh my goodness, this is not how it's supposed to work. And again, imagine we're doing it as this project that, people are definitely paying attention to. What if I was, right, I was a grad student. I used to have to ask people. Well, that seems like a very inefficient way of doing it. And then what, one person decides if a reagent gets to get used again or not, even though, again, it's public funds, they're done using it, even though they want to keep researching it. Why can't they let others? We'll make more progress that way. But that's a challenge for us. So I'll give a good example. We couldn't even replicate one because a lab refused to share us their like mouse line that they made. It was like a quadruple knockout mouse. So don't get me wrong, it took them years to make. They were like, oh, just make it again. It's like, That's crazy, right? There'd be like millions of dollars, years and years of work just to create the reagent, let alone do the experiment. So these were huge barriers. And this also shows us just like we're not set up quite yet. And I think there's big improvements on the data front, but I think the material front needs to catch up. Another barrier, and we just talked about it, is the protocols. We always had to ask for information, and we did code it, right? Sometimes they were, and again, like it's hard to know because the devil's in it. Like I said, devil's in the details. You don't know, but sometimes the protocols would be pretty specified. But we were always had to ask questions because there were always assumptions that you'd have to make. Sometimes we were like, okay, well, we don't know if it matters, but there's a whole bunch of little details that we're missing. Can you help us out? There were definitely examples of papers that lacked like key information where we had no idea what was going on. So we specified that out. And then again, like you said, you noted, we had authors, they'd either help us, and actually it was quite, one or the other. They'd either be very helpful, very, very helpful, by the way, or they'd tell us to go away or not respond to us. So it was very little in the middle. It was either super helpful or not at all helpful, by and large. So those are like a lot of the challenges, because like we have to make it through all of those in order to get to a paper that you can actually then have everything in place to replicate.
Yeah, it's a kind of like what brought you into kind of this role and being part of this project. Like, what motivated you to do this? Because for myself, and not to speak for you, maybe for this is a little bit telling, being who I am. I did grad school and I did sciences primarily because like every good Asian person needs to be a doctor. So that was my reason to have to go. That's going into this. But then you find out, oh, I'm actually pretty good at this. I actually like this type of research. I like doing basically these type of analyses where you go in and you figure out, find some sort of underlying truth. Right? What is your story to kind of getting to where you are today? Because I assume a lot of people going to grad school, at least a lot of my my lab mates, when a lot of them want to be professors. Basically, everyone wanted to be a professor. That seemed to be the main career people wanted to be. But so, what led you down this path?
Yeah, so there's two kind of big things that led me down. So it's definitely not a path I was aiming for what that's worth. It was something that just presented itself. Center for Open Science had started in the town I was in, finishing my PhD at the same time, and I kind of stumbled upon Brian and rattled and lost train of thought multiple times. And in the end, he was like, do you want to work for us and do this project? And I was like, yes. But the reason any of this was interesting, I think, is like kind of, like I said, two things. One is those Bayer MGen reports. Like I remember when those first came out, I was in grad school, and those really caused a huge stir. And it was so much finger pointing. Like that's what everyone thought it was, right? It was, and I've met the folks that are on the authors on that, and like good for them for doing it. By the way, they stuck their neck out there pretty far and got some pretty, some threats and other things that happened, which is also very sad to hear. But that caused an alarm because it was like the first time someone was willing to write in like the Wall Street, journal as well, right? There is a problem. But the problem that I think I had, and especially in the academic space, was everyone was like, oh, industry's just pointing the finger at us. And I was like, are they pointing the finger at us? Are they just actually telling us what's going on? So I found it like a hard problem, right? Which is like, what is going on? And the reason like I think I found it more hard is I was having problems myself, right? Like I in my grad work, at one point I had to replicate, I decided to replicate, right, a key experiment of a finding that was just published in the lab I was in. So I was going to follow up, do some cool experiments afterwards, but I was like, well, step one, I've got to make sure I can get the same result. So let's do it again. I spent months and months in the lab. Now, this is really important, right? I'm in the lab that did the work. I started again with the paper. The paper had, just like I told you, it was just like the state of the art. It had no methods. It did the whole thing that I just told you. Had a reference that went back in time about 20 years. And I was like, is that really what you did? No, that's not what you did. You're just referencing the paper that roughly explains what you did. So again, I'm in the lab. So now I'm like uniquely positioned to have this information. It's not public. I went through all the notebooks. I actually had the person. I could talk to the student that, well, at that point I graduated. So we would sit down. And at one point we were talking about like the way I pipette. And I was like, oh my gosh, it can't be the way I'm pipetting. Like, why would that matter? So I eventually, I think 6-8 months later, eventually was like, okay, I'm going to give it one last shot. I'm going to throw everything at it, all the variations I can to see if I can figure it out. And I did. It was one way that this cell biology experiment would work. One way. And I was like, oh, I finally figured it out. I found out all the secret ingredients that no one else in the entire world will ever be able to figure it out because like I had access to resources and I had to do it myself. And then two, I was like, what? It's only one way? This is really disturbing. Like, is this what I should be doing going forward? I'm not so sure. Why do I want to base it on like, the type of, material I'm using to like permealize cells? Why does that matter so much that only one way does it actually give you the result that was published in the paper? that experience, which is, I think, kind of similar to what you were just trying to tell me from your end, right? That really opened my eyes to like, wow, we are really inefficient as an enterprise, right? Like the research process is, I always knew it was hard. I actually like it being hard, but this seemed like the hard stuff that I didn't want to do, which is trying to repeat, right? It's spending months and months. And then, again, Bear Engine came out. I was like talking to other students. I was like, oh my gosh, I'm not the only one having this problem. And to me, I think that this opportunity just motivated me to be like, yes, okay, cool. So let's figure out what is going on. And again, I think doing it in the open was the most fun thing, because it doesn't just show you what the challenges are, it also shows you what's working. And I think people, we tend to forget that in the narrative. I actually find that more interesting, which is we did replicate results. This whole process worked. There's a lot of really cool research that we were able to just pick up and do it again. And that's awesome, right? That's confidence. That's what you want. The problem is half the time was the reverse, right? I can't get the same result. So anyways, I don't know if that makes sense, but that's essentially my origin. I still actually find that super, really, really important and really interesting.
No, I'm glad. I was just wondering if there was something in your childhood that made you somewhat of a truth seeker. I don't know if you wanted to be Batman or something along those lines, right? Going down the...
I mean, when I was not Batman, when I was so my mom would keep track of these little books of when we were little kids of who are your friends, what are your favorite colors, and one of them would be what you wanted to be when you grew up. And I think when I was in kindergarten or pre-K, I wanted to be a fire truck, not a firefighter, a fire truck. So I just tell you that because the next year I wanted to be a scientist. And after that, I always wanted to be a scientist. So ever since I was in either kindergarten or first grade, I decided I wanted to be a scientist. And I did that. I have no idea why, but I moved from fire truck, and specifically a yellow fire truck, to scientist in 1 year. So that must have been the pivotal moment, right? Which is realizing I couldn't be this inanimate object, that I had to be an actual, that's when I guess maybe I was seeking the truth.
Well, that's awesome. I always tell people, when people ask me, "Oh, what did you want to be when you grew up?" They always want to be in sciences and a scientist. I was like, "No, you know what I want to do? I wanted to play in the NBA." But then I figured out that I'm not talented enough, nor am I tall enough, nor or have any of the athletic capabilities, or work hard enough to do that, right? So, it's kind of funny how things come. But actually, going back to what you talked about with incentives, like this, I'm curious what you think about this overall concept. And you can definitely talk about the incentives that you're moving people towards to change things, because for sure, for long-term stability, incentives matter so much. And there's a phrase that is commonly used in academia, which is publish or perish, right? Which is probably terrible, but it's somewhat the truth. And you can maybe even explain what that means. But what do you think about publishing possibly negative results? Because we only publish positive results, but it's very rare that we publish negative results. And at least from my perspective, you actually learn a lot more when something doesn't work than when something works. When something works, it's like, why did it work? I don't know. It could be a whole bunch of different factors. But when something doesn't work, you almost know instantly what was the thing that made it that it didn't work. But we don't actually publish any of the data. So, what do you think about that component, about maybe publishing negative results? And then, of course, the other incentives that you and your team are proposing, or at least driving towards.
Yeah, that's a great question. Yeah, let's go back to publish or perish, and a little bit of still, to be honest, we're trying to change it, but it just takes a lot of time. So one, scientific papers are by and large positive findings. They show, and I think if you think about it, it's like a nice story, right? I think we as humans like stories, and this is the part of science that is storytelling, right? You're not making up the story, you're trying to show the evidence, right? There are some people that make it up, but we tell it like a story. And the reason why is we like to see relationships. We like to see patterns. That's kind of, I think, because we're humans, we're wired to do that, right? We're wired to do pattern recognition really well. And so I think we tend to just appreciate that. And the scientific literature is super biased towards positive results, crazy biased, right? To the point where you're really, if 90% of all research was, confirmatory, oh, I found this, why are we doing it then, right? Clearly we're just brilliant. We just know everything. But you're right. It's not what's happening. What's happening behind the doors that we're not sharing is actually there's many times it did not work. And those are incredibly important. Like I said, if it doesn't work because I dropped something, that's just a mistake. But if I did everything and my controls worked, you're right. It's incredibly powerful. But before we get into I think some of the incentives to show that, the challenge we have is the literature is largely positive. To get published, you have to have these positive findings, this positive narrative story. Lots of experiments that are all positive, show some relationship. If you don't proceed, right? And so if you think of, money is our true currency, but in science, papers are the currency. So if you want to succeed and stay in, especially in academia, you need to publish papers. You need to publish papers to get a job. You need to publish papers to get a grant. And you need to keep publishing to show you're productive if you want to stay in any of those areas. So you get a really, you publish or perish. If you do not publish, thus if you do not get positive findings, you go away. You will not have a career in academia. At some point, you'll not get a grant and you'll not have a job. This is probably also just slippery. This is like a slippery slope. This is probably, why some people, I think, do slide all the way into fabrication, because if I need a paper and I can't produce one when I'm doing research and I'm about to lose my job, the incentives have essentially forced me to do a behavior that I think is wrong. And I'm guessing, I have no idea why people do that, but I can see that we're leading them that way. So how do we get away from that? So to me, it's all about rigor. We as scientists, we do not control outcome. If we did, we wouldn't be scientists, right? We'd be, just story writers and doing other things, or be back in the day of snake oil and stuff like that. Can make any claim, but instead we control the design. What is the question I'm asking? How do I approach it? Right. That includes all the things of what's my experimental setup, right? All the conditions I choose, my model, all of those are completely in my control, because I'm choosing those, including how I analyze the data, to approach a really important research question, a hypothesis. But I do have zero control on it, because if I have control of the outcome, again, it's completely wrong. Yet I just told you the whole system is wired towards outcome, the thing I do not control. So I think publishing negative results, null results, we'll say, right, those are really good. People have tried making null journals. I think they're a good idea. I don't think they work. And the reason they don't work is because they're, by definition, they're kind of considered oh, right, you're an inferior product to what everyone else wants. So what's more interesting that we've been doing, and actually I used it in the project with the journal eLife, who is publishing all of this. So if you go, you'll see every single one of these. And it's a publication format called registered reports. So what registered reports do, and it works really well for our replication, is instead of submitting, so we'll go back, we'll say, how does the normal process work? So normally as a researcher, I have a question, I do some research, I eventually figure out, I think I have enough to write a paper, I write my paper, I submit to a journal, right? All outcomes, it goes, if I'm lucky, hopefully that journal accepts it after a bunch of revisions, and then I'm done. So the peer review process is way at the end. It's after I've done all the experiments, and they're looking at the outcomes and supposedly looking at the methods, but the outcomes are right in your face, so you tend to focus on that. Registry Reports moves that process to earlier. It's saying, okay, well, you have some pilot data, maybe. You have an idea. So replication is easy because I don't need pilot data. I actually have the original finding. And you say, well, what are you going to do? What is your approach? What methods are you going to use? What materials are you going to use? How are you going to analyze that data? So the peer review occurs before you know the outcome. It's all about what's your question? What's your design? How are you going to analyze the data? And so the beauty of that is when you get that accepted, the journal agrees, they give you in principle acceptance to publish the outcomes, the final paper, no matter what happens. And so now you have a better, you don't have this biased literature anymore. Now you're saying, listen, as long as the rigor is there, right? It's a good quality question. You designed it right. The outcome is the outcome. And so whether it's positive, great, or whether it's negative, it doesn't matter. As long as the controls work out, you did the right thing. So this is actually what we did. This is why we published every single paper in this project is, I think if we did not use this format, most journals would say, I'll publish your positives, but I won't probably publish your negatives. Or I don't know, maybe replications are the opposite. Maybe they'll only publish it if it's negative and not if it's positive. It has a reverse bias. I have no idea. But we instead chose this registered report process because I think this gets at a better way to show what is occurring at large. I'll put it in a different space too. Your example was a really good one of I did it, one out of every 10 times it worked. There's a different one too, which is to realize if I think of Edison making the light bulb, right? He failed 100 times before he finally found the one that worked. But if you think about it, you're absolutely right. We focus on that, and we should, by the way. I think that's great. But every step, every failure was actually a step towards success. So if we don't show those failure steps, we'll actually, I think, collectively never get to a success, because science is not an individual person. It's actually a collection of researchers. So we need to share what's not working so we can collectively get faster to that light bulb.
Absolutely. I'm on the same page with you on, at least conceptually, that this is a collective. This is a team sport in science, right? We're all in it to win it together, because it's any progress that's made is actually beneficial for all of humanity. But obviously there are egos in play. There are quite frankly, there is money involved as well. The more tenured, longer tenure you are, I'm sure you get paid a certain amount as well. So there's definitely those components in play. And then the other, like, how do you adjust those components? Because like there's register report process, it's very, very interesting. How do you get every single journal to adopt this format? Like, how do you get like a Nature or Science? I mean, these are, I don't know if I should be calling out these journals, but they're just very, very high. Let's just say any high impact journal, right? If you got them to adopt this philosophy, it seems like that would what would force everyone to kind of go down that road.
Yeah, it's a great question. And there's also an open question, just like how replicable should all research be replicable? I'd also argue the same thing here, which is, well, does all research have to go through registry reports? Maybe, maybe not. Like, to me, it works really well for confirmatory research, but I actually am fine with people just saying, like, I just have a bunch of data. I don't know what to make of it, but it's data that can be used to mine. So I think another way to say that is there are definitely cases to not use registry reports, and I think that's completely fine. That just allows somebody to just publish data because they're publish some new interesting data set, which you can still do that, right? When you make the human genome, right? Good example. Not a hypothesis there, it's data. So not everything has to be it. That's one thing. But adoption is low right now. But it's interesting to look at where registry reports has started and where I think it should translate over to other fields in this space in preclinical, like cancer biology or preclinical biology at large, like is ripe for it in my opinion. But it started over in the psychology sector. Again, I think they're a group to pay attention to because they did a lot of these things, and I think it's like, how do we translate them now to a different field? There, you have Nature now that has accepted it. So if you're a social behavioral researcher, you can do a registry report at Nature now. So the trick now, we've got it there. The trick now is to get it so that it's any field. And that's going to take time. So I think this is one of those parts that I find interesting. We need to understand it more. It's, think of it as like anything that comes out of, a clinical trial, we don't stop studying it. It's the same thing here in this space. When we have a new intervention, registry reports, to me, let's treat it like we do any research. So let's study it, let's pilot it, let's test it. Let's talk about where it doesn't work, right? Let's talk about where it does work. Let's get it fit for size, because versus just saying, oh, you have to do it or I'm never going to do it. I'd rather just say, we're researchers. Let's experiment. Let's experiment on ourselves, essentially, in the sense of how we do our process. So I'd like to see more registry reports being, tested and used. And I think, if you had to pick one low hanging, I'd pick, animal research or, anything that's like using like, human-based, organs, or tissue types and stuff like that. Like anything that's like very close to that, like translational tipping point. Those are like, to me, the lowest hanging fruits to say you should use this process because we want those results to be known no matter what happens.
Yes, fair enough. Fair enough. No, that's very, very good. I'm glad. I actually didn't know that Nature was already publishing articles that were doing this process, this register report process, or at least psychology. So that's really that does give us a little, at least me, a little bit of like hopes that this would transfer over to other parts as well. But actually, I have two kind of general questions for you. I don't know which one you want to go down first. You want the AI one first, which nowadays you kind of almost have to ask an AI question, or do you want to go down to open source or open data area? I have two questions. Which one do you prefer?
I want both because I think they're related to each other. So let's do the data, then come back to the AI. I defer to you, but both I see them related to each other.
Okay, because the open data, open data. So what does that mean first? So let's say the raw data or whatever is used for the analysis is actually freely available, or at least accessible. Maybe not freely, maybe not for free, but it's accessible. Let's say, right? And the reason why I bring this up is because there is in that podcast that you were mentioned on, which is a Freakonomics. Like, good on you guys for being on that podcast. It was awesome that you were featured, and on top of it, just contribute how you contributed. One thing that they mentioned, at least in Freakonomics, they actually did highlight the psychology of the social sciences being one area where there was a lot of challenges. And then, of course, they brought up yourselves and your project that you are undertaking with cancer biology. But then they brought up economics, and they said there's something in economics that there's less of this type of issue, primarily because people are dealing with data that is accessible and available. Right now, there's different ways you can interpret that data. For instance, let's say right now talking about inflation is all the rage, but there's like 8 million ways of measuring inflation, right? Right. But the data is technically all there. You can just measure it whatever way you want. Now you can disagree with someone else how they measure it. You're like, oh, my measurement is better than this type of measurement because of X, Y, and Z, but all the data is open already. So I'm curious to get your take on that approach of basically if groups have to open source their data primarily. And the reason why I mentioned this is because the vast majority of research is funded by the government, right? Both in the US and myself, I'm based in Canada, mostly funded by the government. Why can't the government say, hey, we as a public, this is public dollars that went into this, so you have to make it open. That way you can then everyone can do their own analysis and see if, hey, do I say come to the same conclusions, and potentially I can come to other novel conclusions as well.
Yeah, I think you're absolutely right. And it takes, so I find this one, I think you're right. These are good examples to use. You do see it in biology. It's not the spots that we were looking at by design. So a good example of an area where you see lots of open data is the genomics field, right? So I think there's a couple factors that are here. One is sometimes the nature of the research. If I'm using public data, it's easy. I point to it, right? It's like economics, political science, sociology. Those fields do it. At the same note, those fields also do a pretty good job of, when they publish their papers, doing what they call a replication package. They share their data and their code all together so that someone else can do it. So that's how you can do it at scale. The journals have to say, you always share your data. Unless ethically or legally you can't, then you just tell us why, like clinical trials. Right? You can't share your data, but you can tell us how to get access to it so someone else can look at the data. I think that's the right thing to do. Open by default, closed by exception. The challenge I think we see in the biological space, and even in other fields, psychology's actually got the same thing. You get in other fields outside of economics, political science, genomics, is the infrastructure that's needed to do it, as well as the norms. And to me, I really do think we should be unpacking it. So if we'll use genomics as an example, that took some infrastructure to have it so that there was a shared, not just norm of doing it, but you always deposit it and do things in the same way. That's really hard when you cut across many, many different types of research designs, right? Okay, I have a PCR-based assay. I have, a Western-based assay, Western blot-based assay, an animal-based assay. So do I have to figure out those formats for each one? I know the NIH, for example, is working on this, which is actually really exciting. We're actually helping play a role in this. It's called the Generalist Repository Initiative, so GREI. And it's a way to try to begin saying, okay, well, we invested, rightly so, tons and tons of money in genomics or other big omics data, but there's a ton of other data out there that's not being shared, that could be shared. How do we support that from an infrastructure, a tool perspective? So I think that's a little bit of it. But I definitely, and this maybe, you're about to get to your AI question, and this to me is the real reason why data sharing is important, is it's not just what you said, which I think is very important, probably the most important, low-hanging fruit, which is, let somebody else look at it, reuse it, and reanalyze it, by the way. There's been people who've reanalyzed clinical trial data and gotten different results, right? It's really important to allow the community to keep beating it up. So that way we don't just take one single paper, one single approach and run with it. But the other one is kind of like genomics. You can eventually start to do more when you have lots of data. So there's this value proposition that I kind of think about, which is, wow, right now we want to do more data mining. So, lots of companies exist, lots of individuals are doing this, which is, let's scour. Well, we got all these LLMs, right? They're taking our information and they're doing something else with it with generative AI. We want to do that in the scientific space. And we've been doing it for a long time, predictive modeling, right? Okay, how do I figure this out? In order to do that, I need all this data, and I need it unbiased, right? I need to do what we were just talking about. I want it as raw as possible. So I think you get this weird value proposition that's going on in the field, right? Which is, I don't need to share my non-genomics data because nobody knows what to do with it. And who really wants to look at my mouse survival data? Well, one, yes, you should, because people do want to look at it. But two, if everyone started doing that and you started getting richer and richer data, you could start to ask questions that were never possible before. 23andMe is an example, right? 23andMe started off doing SNPs, right? So here you're like, oh, here's some information that you know about me. You get to the point where you have so much data, you can start doing ancestry. That's how that stuff happens, is you layer on possibilities that were never possible if you were never beginning to share individual pieces of data one at a time. So I personally think it's great, but I do know that NIH, the European Commission as well, they're trying to do the right thing, which is you need to make it possible. So way down low, create the tools, create the infrastructure, create sharing. And then as you start to do that, then you can start to reward that. You can start to make the data even richer with better metadata, right? Other descriptors. But yeah, I completely agree with you. It's like the lowest hanging fruit. I think, like I said, I think the challenge there is just there's not enough resources yet to really embrace the like messiness of this data.
Yeah. I'm wondering because I completely agree with you. I'm on the same page as you, completely aligned. How do you get there? Because I we're just not the greatest. I would say scientists are not the greatest at promoting like themselves nor the projects, right? How do you get more buy-in? Because I think ultimately getting the word out there, hey, you know what, this is actually for the collective good. If we
Right.
do this, this is good for everyone. How do we do that besides mandating it, right? Like NIH, we fund all this. If you take NIH money, you have to open source it. If you don't, then don't take our money, right? Like how do you move beyond, let's say, the stick, or do we have to use the stick and there's no carrot really?
It's a good question. Yeah, no, and it usually, stick and carrot tends to work a little bit better if we're going to do top bottom up. Um, so on these, I think there's little things you can do. There's little signals that you can sell. So I'll give you an example of a good signal. And then if there's anyone who's an aspiring researcher or a young researcher, I'm going to give advice. It's something I do myself that I've watched other people do, and I think is brilliant. The signal is, and NIH did this recently. There's other funders that do it. I just use NIH just because I know that their policy is a little bit better. They changed not that long ago to allow you to post, basically to acknowledge preprints. So preprints are papers that have not gone through formal peer review. So you used to never have to do that, right? So this published perish culture was really bad, right? Like I could have had a paper and I submitted it to a journal. Maybe I'm a grad student. It's my big paper. And then maybe it takes me a year plus before it finally gets accepted. I can't tell anybody, I can't show that on my CV. If I want to apply for a grant, I can't put that down. So basically what it means is like you're not able to show these other practices. They then changed it to say, no, you can put down a preprint. You just have to call it what it is. But yeah, show progress on your stuff. And you know what? Don't even just show us your papers. Show us your cool data sets. And I think that's the weak signal. It's not requiring it, but it's incentivizing it, right? It's saying when you apply for a grant, or institutions are starting to do this, when you apply for a job, don't just tell us the papers that you published. Show us the other cool things that you're doing. So I think that's like a weak signal that you can do. In fact, if you do more and more of that, you can start to like expose more of these artifacts. And the reason why is if you don't ask somebody, how are they ever going to know to show it, right? So you could require it. That's the full stick, right? I'm going to mandate it and I'm going to enforce it. That's a really hard, by the way, because then you have compliance issues. How are you going to enforce this? But instead, I think you can just incentivize it. Hey, why don't you start showing this as another thing on your resume? We'll look at it, right? So instead of just giving us your papers, show us other things. And again, they're slowly doing this. And I think that's one way to do it. So now I'm going to give you my advice that I think is completely tied to it. And I'd love to see it, which is when on my CV, if I start putting down like, I'll do this for both talks and papers. I'll do my traditional CV because I saw somebody do this. It was great. So you have your here's my paper. But behind it, you'll say, and here's my data, and here's my preprint, right? Here's all the other things I did, and you link to it, right? Because we all have. It's the same thing with the talk. Like I post as our center, we put all of our talk slides in an open repository, and I link to each one because I saw somebody else do this, and I was like, this is good for two reasons. One is that's the way I show who I am. I value these, so I'm going to show you I value these. And two, if you ever want to look at it, I made it just a click, just a little hyperlink. Now you can go and see my data. You can see what I'm actually doing. Versus just, you got to go to like the Nature website and find my paper. So I actually think this is like the way to highlight it. Use these open practices to your advantage. Make yourself get distinguished and don't wait for the paper to get published at a journal. Instead, show everything along the way because, you're essentially building your career a lot better that way. So I think, again, I wish somebody would have told me that when I was a grad student because I would have one, done those. And two, it would have been really refreshing to say, oh, I don't have to wait for my paper before I add it to my CV. I can show you what I'm doing in the interim. And I'll just add the paper when it's finally published.
No, that's absolutely great advice. And that if we had to do a podcast on advice for grad students, right? That's a complete different podcast and another episode that we need to do. And it's honestly like probably at least an hour, if not 2 hours. But yeah, absolutely. Project management would be like another one I would completely put up there. But now I'm a little bit diverting from our topic. But on the AI piece, because I don't want to take too much more of your time. I mean, you've been very generous with your time, and it's been incredibly exciting just to kind of have a chat with you. And I've just been admiring your project for a while now. But in the AI front, it's just because it's just a hot topic now, right? And I have two kind of like questions. One of which is, do you think this is good? Does it make your job easier actually, or your group's job easier now with these tools? I don't know if you use any of the tools to help you identify, papers or flag certain papers or flag certain things. And then the other point of my kind of the AI question is, do you worry about, I don't know what the best way to put it, but like deep fakes. Like I don't know if this is something even worthwhile to worry about, but we always hear about, oh, you can have someone create a video now, animation of some very famous person saying words that they did not say at all, right? Like, is there a concern with that for basically in science that people like basically could just make up data or make up data that's so realistic that it's hard to kind of it's hard to decipher, right? So what do you think about that? That potential challenge?
That's a good one. Yeah, we have conversations in our organization around it. I know there's a handful of people that do use some different tools to help with like getting like a first draft of something, right? They'll use different LLM models for that. I know that there's a bunch of people that use it for help with code. It's actually pretty good for that. If you're like we use open source code, like we use R. There's some good tools out there just to help you figure out like, oh, instead of doing a Google search, you're like, I can just ask somebody, how do I do X? It's pretty good for that too. So I think it has some benefits. Yeah, I think my perspective is yes. I think there is concern. I think it's always been there. There's another concern beyond AI that AI is only going to exacerbate, right? These deepfakes, which is these paper mills. That's another topic that you could get into some point, which is there are groups, because of that publish perish culture, because paper is the incentive, there are groups that you literally can pay them to get a paper, which is crazy, right? Pay somebody and they will put your name on a paper that's completely fabricated. And it finds its way into the literature and it gets cited. And so what's happening is like, we're basically like, again, right? Like being published to me does not give you credibility. It's now you have to sift through it. So I do worry about it, but it's already there and it's been there. We as humans have been doing this anyway. So I think you're right. This probably just makes it worse. I actually find AI more exciting in terms of using it as a tool. We're doing some development. We've been working on a project that was funded by DARPA, which is a agency in the US that's through the DoD. To see if we could develop AI tools to help assess credibility, right? So right now, we as humans do it. You're like, well, can I have an AI assistant help me out? So I think there's actually potential for that, basically to help you sift through it. And people have been using, for better or for worse, AI tools to help detect plagiarism, right? Now there's this, you start to get these circular loops, right? Which is like, oh man, is it just like they're creating fake data and you have to find something to catch the fake data? I don't know. We probably are already in that world and we can't tell. I also think it can help you with, exposing more of the research process. We can use it to help do better research. So I'm actually more excited about leaning more into that potential, and just recognizing like, right, you want to stop this, you have to really shift the incentives and you have to open up more of the process. If I want to figure out if somebody made up data, the best way to do it is to say, well, great, show me all the ways that you collected that data. Because unless somebody's going to truly let somebody fabricate end-to-end research process, which that would be dangerous. You're probably going to figure it out if you see more of the process. But if you're only focused on the paper, it's really easy to get tricked. And I think that's the challenge. So the more that we can expose more of the research that's behind the paper, right? The 10 times somebody did the experiment, not the one time, all the methods instead of just a summary of the methods. I think this is how we're going to help correct this kind of infiltration.
Yes, no, I actually, it's exciting to hear that you think there's a lot of the utility of leveraging artificial intelligence, especially in assessing credibility, because this is a concept that I always kind of thought about. Is like, now that, let's say you have tons of results that are negative, right? And like what they are, right? And one person, it's hard to read all these publications. That if you just had AI kind of go through it, now it understands what is like not going to work. If you kind of type in sort of experiment, a protocol, it says, hey, you know what, just flagging this for you here. This potentially, it looks like this has only a 5% of success. Like, yeah, you could do it, but it looks like there's only 5% of success potential, right? But then that goes back to the fact that you need a lot of open data to actually make that actually happen.
Yeah, there's been a lot of absolutely, you need a lot of open data and a lot of unbiased data, right? So I think like this is actually back to the big call, right? Like I find mining AI tools, like I find this super fascinating. Yeah, there's definitely like a con to it. So I'm not like, and there's biases that they can propagate, by the way. So like, don't want to dismiss that. We just have to like really focus on like rooting out those biases as well. But the potential seems so enormous to me. But I think the challenge is, unless you get good, reliable open data, right? Like it's easy to like pump the literature, the data into a model and get something out, but it's like garbage in, garbage out, right? You don't want that, right? I think we want to be focusing more on the quality of data that we're putting into our models, so that way we can actually use them efficiently. So I'm excited for both ends. I see the potential that's there. I still think at best, it's really more of an assistant, right? AI is not something that is going to go off and do it, but that day might change. But I think right now, in order to really see the potential of what it can do for us, we need to like make our data more reliable and more open.
Absolutely, I completely agree. Maybe this end off with maybe just this last question. Don't want to take up any more of your time. I know you have a lot of dad duties now as well, right? So, what do you hope to achieve in the next, near term? You can say midterm, whatever time scale you want to do. Like, what do you want to be able to achieve here at the Center of Open Science, or at least for this Reproducibility Project? What do you hope for in the next, maybe say 5 years?
Yeah. I think that is so. It's a really good question. It's a hard one to answer. So, the big thing I think we're trying to accomplish at the center, and I'll give you then my personal like tilt on it, is really trying to see if we can catalyze more change within communities. So, as we've talked about this whole time, right? It's wanting more open data. It does not work just to say, hey, researcher, share more data. I don't actually like that. I think those become like unfunded mandates. They're really hard. You have to fix the whole system around it, right? It's really, that's it. You have to create the tools, the rewards, the incentives, the policies if you need to, in order to the training, right? That needs to be set up so that way the researcher is rewarded for it. We're humans. We do what we're incentivized to do. We want to be rewarded. We want to do good behavior. So I think my focus, and I think our center's focus, is really trying to focus more on that. Really trying to have strong use cases to say, can we start to like work with these kind of ecosystems to really begin to move the lever on it. Again, I'm really hopeful because I've seen in the past couple of years a lot of change. Right, it's a year of open science that has been declared. NASA's doing a huge role now. White House has been supporting this. So there's a huge push for it. So I think now the trick is, how do you take all that excitement and put it into beginnings of action? So for us in the next 5 years, I think we want to see stronger use cases of how you can shift the culture. To me personally, I want to see that in the discipline that I did my PhD in cancer biology for a reason. That was the one that still excites me today. I find that the most exciting. I love what I'm doing now, by the way. I find that even more exciting. I did not know that, this meta-science stuff. But I still am drawn, and I'm always attentive to that oncology, preclinical oncology field. It's just always, I find it so fascinating because it's such a hard problem. So in the next 5 years, I would like to see something like registry reports, for example. I think that is a game changer. I'd like us to see if we can do something that is going to move the needle on how we conduct research there, that I think will actually have and actually see maybe a potential change in, instead of just doing things status quo, I'd like us to see a change towards increasing rigor or open in this space. That's part of the rationale of doing this project over the last, 8 years, is now I want to say, great, the conversation has shifted from, I think we have an issue, to what are we going to do about it? I think in the next 5 years, I'd like to start seeing more and more groups do that. And by the way, there are groups doing it, which is really exciting, right? I'm just like, next 5 years, I'd like to see more and more of that, because I think there's still some low-hanging fruit that hasn't happened yet.
Great. Hope to have you back on, and you can discuss it when some of these things happen. Hopefully, maybe shorter than 5 years, maybe a year or 2 years, or hopefully much quicker than that. So, thanks a lot for joining us today, Tim. Very much appreciated. Do you want to let the audience know where people could maybe find some of your works? I did already mention that your website is www.cos.io. Any other place that you would like to mention, like a Twitter or some other repository you're interested in mentioning?
No, that's actually the best one. If you go there, and it's if there's a couple, you mentioned one, the /RPCB is there. If you do want to see some of the stuff that we're doing in the AI space, the credibility space, you can do /score, SCORE. That'll give you some more information. And the same thing, if you look around on some of the headers, you'll find stuff on registry reports. You can even just Google registry reports, COS. It'll take you right to the landing page. I think I find registry reports really fascinating. So it's definitely something I think the audience is unfamiliar with it. There's a lot of good, useful information there to really share it.
Great. Again, thank you very much, Tim, for being on the podcast today and sharing an incredibly important topic that we just don't talk enough about really in overall society. And thank you again for all the great work that you and your team do. Thank you very much for the audience for joining us today again. This is, of course, a little bit deviation from our usual topic and my co-host, but I think this is a fascinating topic that not only is actually as impactful as like what we don't really talk about real world data, but really actually aligns very well of at least our philosophy, as well as making sure that the data is available so people can actually assess it and actually do great work with it, especially deriving insights. So thank you again, everyone, for joining in today. If you have any comments, please comment below. If you have any questions, comment below as well. And of course, like and subscribe and follow us. Thanks again, everyone.